Good afternoon, everyone. Welcome to this symposium on animal consciousness beyond mammals. I can hear it. Okay, maybe I'm too short. There we go. Is that better? Okay. I'm Sid Johnson, and my colleagues here today are in order of presentation: Jonathan Birch, Andrew Crump, Tilda Givens, and Peter Godfrey Smith. We happen to be going in alphabetical order purely by accident. I'll be speaking last and also moderating for at least part of our time. Our topic is animal consciousness beyond mammals. And our question is, how should we study consciousness in non-mammals scientifically? And that should is both epistemic and ethical, because the neural basis of consciousness in non-mammals is likely to be significantly different than it is in mammals, resulting in some serious theoretical and methodological challenges. But answering this question also has great ethical and societal importance, since evidence of consciousness or sentience is generally taken to justify welfare protections. And non-mammals currently are frequently excluded from welfare considerations in the various contexts in which they are currently used, including in research and as food. So here's how we're going to proceed today. The first three speakers, Jonathan, Andrew, and Tilda, are going to discuss the study of consciousness in insects, with special attention, excuse me, to bees. And following those three talks, we'll have 20 minutes of Q&A. So please save your questions for that break. And then following that will be the last two speakers, Peter and myself. Peter will be talking about phylogenetically distant organisms, and I'll be talking about ethics and fishes, followed by another 20 minutes of Q&A until the end. So our first speaker is Jonathan Birch, and I'll let you take it away.
Thanks so much, Sid, for organizing this session. And thanks very much to everyone in the audience for staying till this late stage of the conference. It's really appreciated. Thanks too to the organizers for organizing what I think has really been a landmark event in the history of consciousness science. And I'm delighted that as part of that landmark event, the organizers wanted a symposium that was trying to broaden the scope beyond humans and beyond even mammals. I'm really thinking about invertebrates in this talk. I think my interest in this topic was to some extent stimulated by this event in 2012, the Cambridge Declaration on Consciousness, at which various neuroscientists, including Christoph Koch and Jaak Panksepp, got together in Cambridge and signed up to this declaration that said non-human animals, including all mammals and birds, and many other creatures, including octopuses, possess neurological substrates complex enough to support conscious experiences. They were trying to create this moment where we say the science of human consciousness has now reached a stage where we can be ambitious about addressing the big questions of consciousness in other animals as well. I think that was a really important, very welcome thing that they did. And 11 years later now, I think it's fair to say a field is emerging. Think of the journal Animal Sentience, founded by Stephen Harnad, running now for 7 years. A field of animal sentience research that combines a very wide range of disciplines, including neuroscience, comparative psychology, philosophy, my own discipline, evolutionary biology, economics, law, animal welfare, and veterinary science. It's a field that faces foundational challenges because many of the methods we use in human consciousness science rely quite inherently on asking humans to report their experiences. Even the ones that don't often rely on quite specific features of the primate visual system. So the question of how we should go about studying conscious experience in non-mammals with the same rigor we bring to the case of humans, primates, and other mammals is an absolutely huge question and the central one of this symposium. Since 2020, I've been fortunate enough to be building a team supported by an ERC grant to address these questions. It's called the Foundations of Animal Sentience Project. Here's a picture of the team at a snapshot in time in September of last year. We had Andrew and Tilda, who you hear from later, Daria, who gave a talk this morning. Also, Joe Ledoux was visiting us at that time as a visiting professor. I see this as a sign of our commitment to trying to win over even people who say they're not interested in animal sentience when you ask them. And Joe's made some pretty valuable contributions. Our goals are to develop better methods for studying the conscious experiences of animals scientifically, and new ways of using the science of animal sentience to design better policies, laws, and ways of caring for animals. One of the key papers I've published in recent years on this, one that I think captures my outlook on these issues more accurately than any other paper, I think, was called The Search for Invertebrate Consciousness, where I argued for what I call a theory-light approach to these questions of consciousness in non-mammals, including invertebrates, which starts from the thought that given the extent of theoretical disagreement in the human case, which is very significant and I think has been exemplified throughout this conference, it would be a mistake to premise the entire field of animal sentience research on a huge bet in a particular theory being correct. I think that would be a mistake. We need to avoid premature theoretical commitment. At the same time, it's crucial that we must move beyond credulous overinterpretation of human-like behavior. In one of the commentaries on one of the articles I published, someone said to me, well, how can you even doubt really that a scallop is conscious when you see the way it swims off when attacked by a starfish? And I mean, it's hard to know what to say to this sometimes. I mean, I can doubt the possibility of conscious experience in scallops. We need a rigorous science of animal sentience that addresses these questions through experiments and evidence, and not YouTube videos. How do we do this, though? Points of wide theoretical agreement is where I go in this paper. I argue that points of wide theoretical agreement behind which the field can unite can help us identify the relevant similarities that actually provide useful evidence. And I suggest in the paper that here, this can be one point of wide theoretical agreement, what I call the facilitation hypothesis. The hypothesis that conscious perception of a stimulus facilitates, relative to unconscious perception, a cluster of cognitive abilities in relation to that stimulus. That leaves room for disagreement about what those abilities are. It leaves room for disagreement about whether that's all that conscious perception does, whether sometimes it impairs cognition, for example. It leaves room for disagreement about whether this is part of the essence of conscious perception, whether it has a functional essence, or whether this is something it does contingently in the way one might think if one has a local theory of conscious experience, that it's a sort of perception that facilitates downstream processing, but not essentially. I think that all defenders of current popular theories can get behind this idea, and that this idea is rich enough to guide a theoretical program of inquiry into consciousness in non-human systems. A program that I call the theory light program because it is not completely theory neutral, but aims to structure itself around a minimal theoretical commitment that a wide range of people from across the field can support. Step one is to use human and mammal work to find cognitive abilities that are at least plausibly facilitated by conscious rather than unconscious perception. And the session with Lena Scora and Ryan Scott the other day was a great example of that kind of work. Step two is to then use that as a starting point. Try to translate the paradigms through which people have produced these dissociations in the human and mammalian case, and look for similar patterns of facilitation in a wide range of other animals. Crucially, we're not just looking for the abilities. I'll give an example in a moment, but we're not just looking to see how good animals are at learning or how intelligent they are. We're looking for a pattern of facilitation of certain functional properties by conscious perception that is similar to the pattern of facilitation we find in our own case and in other mammals. Step three is to then look for a distinctive kind of processing that is responsible for these patterns of facilitation and use that as the basis for an inference to the best explanation, an inference to the hypothesis that this distinctive kind of processing we found, whatever it turns out to be, is conscious perception and that it is present in this other animal. I think that various labs around the world, including my own group, including Lars Chitka's lab, with which we've been collaborating closely, but also various other labs, Ralph Greenspan's lab, Martin Djurfer's lab. People have been thinking in this way implicitly already. And I think it's time to coordinate and think of this as a unified research program that can give us a serious science of animal consciousness. I'll zoom in on one particular example that illustrates this approach in action. It's the example of, oh, by the way, the slides for this talk, if you want to bring them up on your own screen, as it says here, just go to bit.ly/birch ASSC, or you can scan the QR code. And as a little incentive, if you do that, you get this nice bonus slide in which one of my postdocs, Simon Brown, has crystallized the Theory Light program into a nice diagram. But I won't go through the whole diagram now. Instead, zoom in on a specific example. This is one of the best known putative dissociations between conscious and unconscious processing in our own case, the example of trace conditioning. It goes back to work by Clark and Squire in the 1990s on humans, but also on other mammals, including rabbits, that suggested a surprising dissociation between a form of classical conditioning where the stimuli are presented together and they overlap in time. It seems to be very simple, supported by quite simple mechanisms, and a more sophisticated form of conditioning where all you do is introduce a temporal gap between the conditioned stimulus and the unconditioned, in some cases a gap of just a second or even less. It seems that introduction of a temporal gap ends up implicating quite different mechanisms because the trace of the stimulus has to be somehow retained for use in future learning. And the key result from the Clark and Squire work is that it seems as though trace conditioning, but not delay conditioning, is facilitated by conscious perception of the stimulus. But also, moreover, that trace conditioning, but not delay conditioning, is very sensitive to distraction after conditioned stimulus onset. If you give people the conditioned stimulus, could be a tone, could be a visual stimulus, then you distract them, and then there's the unconditioned stimulus, which in these experiments was a puff of air at the eye, to which you have to blink at just the right moment to avoid it. This is very sensitive to distraction, and subjects no longer learn it if they're distracted before the air puff. And moreover, they found evidence that the two types of conditioning depend on different brain mechanisms. They found that trace conditioning is badly disrupted by damage to the neocortex and by damage to the hippocampus. Delay conditioning, by contrast, just seems to carry on regardless. This is often said to point to a possible relationship between conscious perception and learning about temporal relationships. Now, none of this is uncontroversial. When is anything in consciousness science uncontroversial? Critics say, well, that difference between trace and delay conditioning may not be explained by the involvement of consciousness in trace rather than delay. It may be that there's confounds, in particular, Haquan Lau, Megan Peters, Matthias Michel, have encouraged us to think seriously about the possibility of signal strength confounds, where if you do something like mask a stimulus or present it for a shorter duration, you make it intrinsically weaker and harder to discriminate from noise. They argue that it's that weakening of the signal rather than the involvement of consciousness that explains the differences in these cases. And we're not going to resolve that debate right now, but I think a crucial point is that we don't need to resolve every controversy in human consciousness science in which to use tentative dissociations as a guide to our research into other animals. Let's see if we can reproduce those dissociations. Let's look at the mechanisms involved and see what picture we end up with at the end of all that. And that's what recent research has been doing. We can ask these questions now about insects without waiting for a resolution of the human controversy. We can ask, are they capable of trace conditioning at all? We can ask, is the trace conditioning supported by different mechanisms from delay conditioning? We can ask, is the trace conditioning mechanism differentially affected by distraction, as it is in our own case? And we can ask, is the trace conditioning differentially affected by masking-like protocols, which of course we will have to adapt to the species. The ones we use in the human case exploit very distinctive features of the primate visual system. Now, some amazing work, including in particular this paper by Dhruv Grover and colleagues from Ralph Greenspan's lab at UCSD in Nature from last year, has been exploring exactly these questions. It's a rather remarkable setup in which they have the Drosophila fruit fly tethered in a virtual reality arena in which stimuli are presented in the inside of a ball that surrounds it. And it flies around its environment, and they train it so that the presentation of a certain stimulus, like an upright T, is followed by a noxious stimulation delivered by a laser. And they can look at delay and trace conditioning in this setup. What happens when the T and the laser overlap in time? What happens when there's a temporal gap between them? How does that affect the learning as manifested in this test setup where the fly faces the conditioned stimulus and another stimulus, and the question is, will it fly away from the one that predicts laser if it goes in that direction? And what they found, I mean, it's fantastic stuff. They found genuinely there are different mechanisms involved, and the trace conditioning, but not the delay conditioning, is sensitive to distraction in the form of an air puff administered to the fly in between CS and US. Moreover, because this is Drosophila, they can do very high quality neural recordings of what is going on in the fly's brain while this is happening. What they found is that there's, and this is from a paper by Martin Joffe, Catherine Macri, they found that the activity that represents the conditioned stimulus, the visual stimulus, in the central complex in the middle of the insect's brain, modifies within just 7 trials, a very small number of trials, modifies to already be firing during the gap to keep the stimulus alive during the gap in time. But moreover, they found that also the representation of the unconditioned stimulus in the form of dopaminergic neurons. Really amazing thing about insects is that dopamine is a punisher in insects. They hate dopamine. They're masochists. And that representation too, also, the neuron also is firing during the gap, as if preparing for the punishment. Just seeing the T starts to be experienced as a punishment, as it were. So the quality of this work is fantastic, and it answers several of our questions. Are they capable of trace conditioning? Yes. Are the mechanisms different from those underpinning delay conditioning? Yes. Is the trace conditioning mechanism differentially affected by distraction? Yes. Is trace conditioning differentially affected by interventions on the stimulus, like masking-like protocols? This is unknown. But I think they're giving us here the sort of setup in which this question could be probed. Similar results too in honeybees from Martin Joffe's lab at Toulouse, where they're exploring olfactory trace and delay conditioning rather than visual. Very striking that the same dissociation has been reproduced in 2 different sense modalities. So no one would claim that this work conclusively establishes consciousness in Drosophila. We're not in an area where conclusive evidence is just going to suddenly appear in an issue of Nature like that. But it is, I would say, weakly probability raising. If you were initially skeptical of there being 2 pathways for perception in insects, one of them conscious, one of them unconscious, as in us, this evidence shifts probabilities in an interesting direction. It leaves many questions open, of course, as well. It leaves open the question of whether we can find masking-like protocols that affect trace conditioning. It also leaves open the question of, can we reproduce other dissociations? The case can't rest on trace conditioning alone, of course. We need to look for other dissociations too, including those relating to metacognition, I think, too. I think it also raises this question of, to what extent are the bees' capabilities, or the fly's capabilities, constrained by the sort of setups in which they're being studied, in which they are tethered, they're glued to a single point, they cannot genuinely move. I think it's entirely plausible to think what we get here, even though it's incredibly impressive, is a very limited picture that is not showing their true capabilities. And a recurring message from the bee and insect literature is that when you study them in free-flying conditions, they're often capable of a lot more. Now, Andrew Crump, in the talk that will follow this one, has been exploring all of these questions, including the one about whether we see different behavior in free-flying conditions. I just want to end with a reprise of the main message, which is that various labs around the world have been thinking in this way already, that we don't need to commit to a single theory to study consciousness in other animals. We don't need to credulously overinterpret behavior either. What we need is to look for dissociations where a certain kind of ability is facilitated by conscious perception relative to unconscious perception. And I think it's time to coordinate and think of this as a unified research program that holds tremendous excitement, because we could be talking here about the discovery of a new model system in which we can study conscious experience, one in which we have very, very good resolution on the precise neural mechanisms involved. So thanks very much for your attention. The link to the slides is up there. And the next talk will be from Andrew Crump, who will take up these themes further. Thanks.
Thank you very much, Jonathan. And good afternoon, everyone. And firstly, thank you to the organizers for the opportunity for me to present here today. I'm going to be talking about conscious and unconscious vision in bumblebees. And I'm going to start off with a caveat, because that's a pretty bold title, that the experiment I'm going to present today, I don't think whatever the results were, could have conclusively demonstrated that bumblebees have conscious versus unconscious vision. What I do think from this experiment is that it provides us with a paradigm where we might start to test that idea. So going forwards, it's sort of the first step on a path to hopefully addressing this question of whether bees have distinctive conscious and unconscious vision. Why does that matter? Well, typically evidence for consciousness in animals like bees, these real kind of edge cases for consciousness where we don't really have any idea whether they are conscious or not. Typically, papers that claim to provide evidence for that start with a single sort of cognitive or behavioral marker. I'm going to focus specifically on behavior rather than neural evidence today. So one example of that, which Tilda's going to talk about next, is motivational trade-offs. So I won't go into detail about that now. But basically, these papers say, can bees perform motivational trade-offs? Yes or no? If yes, that's kind of some sort of weak evidence for consciousness in bees. I guess I see two main issues with that as a line of evidence. The first one is that we don't really have, as Jonathan was saying, this strong link between any cognitive abilities in humans and consciousness. There's no kind of uncontroversial cognitive gold standard marker for consciousness in humans. That's pretty controversial. I welcome disagreements on that point. The second issue is that even if we did have this kind of gold standard marker in humans, even if we had a cognitive ability or a behavior which we knew required consciousness in humans, it's pretty plausible that animals might be able to do it non-consciously. They might have a different mechanism. And so I think this single marker approach, although it is probability shifting to a limited extent, is limited. So increasingly, I think people have been moving towards looking for, as Jonathan was saying, a cluster of cognitive markers, and we can list sort of various cognitive abilities which either empirically or theoretically might be linked to consciousness. So certain perceptual illusions and certain types of learning, as Jonathan was talking about. And here I think we start to make consciousness a more parsimonious explanation. If something like a bee has all of these things, which we accept potentially that in humans all these abilities involve consciousness in some way, then if bees are doing all of it, then suddenly consciousness becomes a fairly parsimonious explanation. But still, I don't think we truly address those two issues, that one, none of these are uncontroversially linked to consciousness in humans, and two, a bumblebee, for example, might have evolved a separate mechanism. And so this brings us, I think, to what I consider to be the potentially strongest line of evidence for consciousness in something like a bumblebee, which is to demonstrate a conscious-unconscious distinction, to show that for supraliminal stimuli, yes, they can do these tasks or they can perform these abilities, but for subliminally presented stimuli, they can't. Because then suddenly you've got this evidence not only that bees, for example, are conscious, and not only that these cognitive abilities indicate consciousness, but that these cognitive abilities indicate consciousness specifically in bumblebees. So that, to me, is why this conscious versus unconscious, supraliminal versus subliminal distinction is really interesting for bees. The trouble, of course, becomes, well, one of the many troubles becomes, how can we validate subliminal stimuli for something like a bee? As Jonathan suggested, and as we all know, many masking paradigms, for example, rely on parts of the brain which bees simply don't have. So a visual cortex, for example. So how are we going to even validate that these stimuli, which we use so commonly in humans and in primates, are actually going to be effective as subliminal stimuli for bees? And so that's, I guess, kind of the aim of my research program at the moment, to try and do that, to try and say, how can we even go about validating potential subliminal stimuli? And to do that, I think we need to think in very basic terms about what subliminal stimuli actually are. So we have on the left-hand side in this figure, 3 stimuli, a dot of different levels of contrast with the black background. So at the bottom, the dot is black, you can't see it at all. In the middle, the dot is gray, so it's sort of intermediately hard to see. And at the top, the dot is white, so very, very easy to see. And so if we're thinking about subliminal stimuli, we have this subjective threshold. And above this threshold, we report perceiving the stimulus, in this case, the dot, and we can also use the stimulus to perform above chance levels in a cognitive task. But then the really interesting point comes here between the subjective and the objective threshold. This is when we report not perceiving the stimulus, but nonetheless can use it to perform above chance levels in a cognitive task. And then finally, we have stimuli below the objective threshold where we report not seeing them, and we can't use them to perform above chance levels in a cognitive task. And of course, the subliminal stimuli fall between these two thresholds. So this is really where we're looking for bees. We're looking for stimuli that the bees report not seeing, but nonetheless can use to perform above chance levels in a cognitive task. And here's where things get really difficult, because as you probably guessed by now, bees can't really report. They can't tell us what they're seeing. Animals can't talk, right? I have a PhD in biology, everyone. And so the question is, how can we go about validating these subliminal stimuli if bees can't tell us, okay, I see this, I don't see this. How can we do that? This is where I think we can usefully borrow methods from metacognition. So as if consciousness science itself is not full of tricky controversies enough, we're now going to bring in animal metacognition research. I use metacognition methods not to provide kind of higher order evidence for metacognition as evidence for consciousness in bees. I use it simply as a methodology for bees to report. Because if we're talking about bees reporting their own perception, that's essentially metacognition. Do they know what they know, and can they tell us? Very usefully for me, there has been work on metacognition in bees before. So this is a fantastic study by Perry and Barron from 10 years ago now. And it's a pretty standard paradigm in animal metacognition research. I will say right from the outset that I realize there are a lot of potential confounds in animal metacognition research, a lot of killjoy explanations. But for the sake of argument, we just need to accept that the results are potentially consistent with metacognition. So in this experiment, the bees were given 2 arenas, each with a task, and basically they could go through arena 1 into arena 2. The task in arena 1 was very simple, just the target, which in this case is a cross. If it was above a reference line, it's rewarded with sugar water. If it's below the reference line, then it's punished with quinine. But importantly, the bees had this exit to the second cylinder, so they could opt out if they didn't think they knew the answer. In the second cylinder, you then have this forced choice version of exactly the same task. And so I like to conceptualize these 2 arenas. The first one is a kind of self-report arena, because if the bee is reporting, I know how to do this, it's just going to do the task in the first arena. Whereas if it's reporting, I don't know how to do this, I can't perceive any difference, then it's going to opt out into the second, where we then get the forced choice cognitive task. I'm not going to go into detail on these results, but essentially they are at least consistent with metacognition in bees. So in the graph on the left, you see that there were more opt-outs if the task became more difficult relative to the easy version. So bees were opting out more for harder tasks, at least consistent with metacognition. And then in the graph on the right, you can see that for the bar on the harder trials, the bees were performing better if they were unforced than if they were forced. So this again is at least consistent with the idea that bees knew whether they were going to perform well. So the aim of our work was essentially to try and use this kind of paradigm to validate subliminal stimuli for bumblebees. So use this metacognition paradigm to validate subliminal stimuli. Our experimental setup was very simple. So we had, was very similar to the Perry and Barron study. We had a sort of nest box with a colony of bumblebees attached to 2 arenas, so bees could opt out of the first one and head into the second one. And this is a top-down view of those 2 arenas. You can see that they basically had a floor which was computer screens. And on those screens, we were able to present stimuli. So in this case, just for training, it's very simple sort of white or light gray dots. So the bees learned to go to the dots for sugar reward and to not go to the non-dot positions, or else they got this sort of quinine punisher. And they could do that both in arena 1 and in arena 2. Then for testing, we basically just made the dots progressively darker, so reduced the contrast, reduced the luminance of the dots, and the task got harder and harder. And then again, just like in the Perry and Barron study, I think of the arena 1 as being the kind of self-report. The bees are telling us if they do the task in that arena, yes, I can see the dots. And they're telling us if they go into the next arena, no, I can't see the dots. Okay, so the hypotheses of this, basically, if we see these distinct thresholds in bees, would be a pattern of results where above a subjective threshold, the bees would get the answer right in arena 1. They'd perform above chance levels in arena 1, and they wouldn't opt out because they would be reporting being able to see those dots. For these potential stimuli between the 2 thresholds, the bees would be expected to opt out in arena 1 to report, no, I can't see these dots, I'm going to try it in the next one, but nonetheless perform above chance level in the forced choice version. And then finally, once we got below that threshold, the objective threshold, the bees would opt out in arena 1, but perform at chance in arena 2. And these, if we go forwards with this experiment, would be potentially in the future, if we try various masking protocols or different types of subliminal stimuli, could then be potentially validated subliminal stimuli. Okay, the results. I'll again caveat this by saying that some of these results are quite tentative. I haven't finalized the analysis yet. This slide is just demonstrating that some assumptions of the task were met. So firstly, that the bees got more correct responses when the task was easier. So when the luminance was higher and the contrast with the background was greater. We also, because I always get this question, especially at entomology conferences, we also did non-reinforced trials. There's been some recent fairly difficult research for some of us who work on bees suggesting that actually bees can smell sugar from some distance away. So we did unreinforced and reinforced trials and we found no difference between the two. So bees were genuinely learning here. I'm not going to go into too much detail on the metacognition aspects, except to say that like in the previous work, our results were at least consistent with metacognition in bees. So this graph shows whether the task was impossible. So the only screen 2 showed the dots, that's the box on the left, or both screens displayed the dots, that's the box on the right. And you can see that bees are opting out significantly more if the task is impossible. That's exactly what we'd expect. And then finally, and perhaps most intriguingly, when we looked for those distinct metacognitive and objective thresholds, we found a kind of remarkable convergence between the two of them. So the bees were almost identical in when they were performing at chance levels and when they were showing this big threshold of increased opting out behavior. At least it seems from this initial experiment and from these initial data that actually there isn't this sort of divergence of the two thresholds, that they're almost identical to one another, which is sort of fascinating that bees show this remarkable consistency between these two thresholds, but not perhaps what we expected or what we really wanted going forwards, but interesting nonetheless. And I guess I'm reticent to draw too many conclusions from this work so far, but one option could be that bees are completely non-conscious. All their perception is unconscious, both metacognitive and objective thresholds are identical. They are, if you like, zombies. Thank you. The alternative. I should say I nicked that from a title of a philosophy paper, so apologies to whoever wrote that. The other alternative potentially is that maybe bees have only conscious perception. I mean, that's at least a plausible possibility, right? I don't know. And I don't think we do know necessarily from these data, but I do think that it's really exciting research program going forwards to use methods like this in order to try and look for potentially this distinction between metacognitive and objective thresholds. So I'm really excited about that going forwards, and I hope you are too. So thank you very much to all of my collaborators, and thanks for listening. And then speaking of my wonderful collaborators, it's my great pleasure to introduce Tilda Gibbons, who is much better at getting bees to do cool stuff than I am.
How do I? It's fine. We got it. All right, thanks. Okay, so I'm going to talk about my research on the neural and behavioral indicators of pain in insects. Oh my God. Okay, I'm pressing the wrong button. That's what I'm doing. All right, there we go. Okay, so first I'm just going to go through some definitions. Whoops. So first of all, a noxious stimulus is a harmful stimulus. So this is basically something that causes harm to the tissue. And in this diagram, you can see it in the corner. This would be like for an injection or a burn or something like that. So you can see it on the hand. So this is basically just something that causes harm. Nociception is then the processing of this stimulus. So this is, in this diagram would be in the neurons carrying it to the spinal cord and then up to the brain. So this is the biological processing of the stimulus. And then pain is specifically the feeling that's generated in the brain that's the negative subjective experience. So I just wanted to define between these two because nociception and pain are conflated quite a lot, but I'm specifically interested in whether insects can feel pain, so the actual subjective feeling that's generated in the brain. Okay. So first of all, we already know that insects have nociception. So we know this because they have channels and nociceptors that detect the noxious stimuli. So they're able to very basically respond to these stimuli. And they also have behavior showing that they're responding to these with reflexes. So for example, Drosophila larvae will roll away from a noxious stimulus and Drosophila adults will jump away. So they have these very quick acute responses that show that they are responding to noxious stimuli. So that's the basic nociception they definitely have. but whether they have pain is the question. So how can we measure pain? Measuring pain in humans is a little bit easier because in verbal humans, we can use these pain scales where they can tell us about their pain and they can say how bad the pain is, they can rate it, and they can say even where it is and what it feels like, whether it's dull or sharp and where it is. So obviously that makes things a little bit easier. In humans that can't talk, we can use behavioral scales as well, which are developed based on what verbal humans say when they're feeling pain and how they're behaving. So if someone is grimacing or is acting restless, we can then assume that they're feeling pain based on what a human that is able to say they feel pain would be behaving in that situation. So we make that assumption, but we're all okay with making that assumption. We also make the same assumption for some non-human animals. So non-human mammals. So for example, dogs in veterinary clinics will use this pain scale where we look at vocalizations and rubbing or licking the area and all of these behaviors that are associated with pain in verbal humans. So this is where we're getting this information and this is why we make this assumption. But we don't currently make the same assumption for invertebrates or insects. But we have got lots of indicators about the nervous system and behavior. And these are just lots and lots of lists of indicators that have been developed into looking at pain in insects. I'm just going to focus on these two particular ones because these are the ones I've focused my research on. So the first one is motivational trade-offs. So motivational trade-offs, when associated with nociception, basically mean that the natural avoidance of a noxious stimulus is traded off with the motivation for something else. So an example of this in humans is how we can endure getting an injection to basically help us avoid illness in the future. So we'll sit through the pain, the small pain of an injection to reduce our harm in the future. So the reason we can do this is because we don't just run purely off reflexes. So we can suppress that reflex. If we couldn't, we wouldn't be able to sit through it and we would just run away from the doctor every time. So this shows that we have some sort of system for modulating based on context, our responses to noxious stimuli. So this is what we were interested in whether insects can do. So I tested this with bombostratius bumblebees. Obviously, the trade-off would be a bit different than in humans. So I tested for whether their avoidance of noxious heat, so it'd be 55 degrees Celsius, this is the one I used, or 131 Fahrenheit, and whether this is traded off for their motivation for a higher concentration of sugar, which is highly rewarding for bees. So this is the setup. If you just look to A, to start with. So basically the bees were offered two different types of feeder. So they were offered the pink feeder and the yellow feeder. Both of these in this condition, both contain the same concentration of sucrose, so 40%, which is really highly rewarding for bees and they really like it. But the yellow feeder had a heat pad underneath, heated to the noxious heat. As you can see in the white bars, they much preferred feeding from the unheated feeder, which would make sense. Their natural avoidance is to avoid the heat. They would prefer to. But when we changed the conditions, kept everything the same, but just changed the sugar in panel B, we changed the sugar concentration in the pink feeder to just be 10% now, which is still sugar. It's still food for bees, but it's a lot less rewarding than 40%. And as you can see in the white bar, their preference went straight to the heated feeder, which means that they're able, based on the context, to start enduring this heat that they would prefer to avoid based on the context. So we also then tested straight after this experiment, we took away all of the heat and we replaced all the sugar with water. This was just to see whether the bees were remembering what was at the feeders and making their decision based on the memories of the feeders. And as you can see from the green bars, these responses were very similar to their normal responses when they could see all of the unconditioned stimuli. So this tells us that they were making these decisions based on what they remembered was at those feeders. And then this graph is just basically the conditions I just showed you at 10 and 40, but we also tested when the sucrose concentration was at 20 and 30%. And as you can see, the preferences as you'd expect, it sort of just goes down as you're increasing the concentration at the other feeder. So basically the trade-off extends throughout different variations. So our conclusion from this is that bees will endure the heat to get the stronger reward, and that they will perform motivational trade-offs. And I'll try and play this video. This is just the bee feeding on the heat pad to get the sugar. Okay. So the second indicator is flexible self-protective behaviors. So this is basically when we protect the site of noxious stimulation from future pain. So an example of this in humans is when we stub our toe and we grab the toe and rub it better, or when we're limping to try and stop future pain, or when a dog is licking the wound for the same reason that we rub our toe. So we wanted to see whether insects do this. So this slide is basically just an example of lots and lots of papers that have said that bees, insects in general, don't do this, and that there's no evidence of them doing this. And because of this, this means they don't feel pain. But all of these quotes are based on a lack of research rather than any negative research. So instead, it's just, we don't have any evidence of it, but that's because no one's actually looked for it. So that's sort of why these conclusions have been made. So we tried to look for it in bumblebees again. So this was our setup. The bees could usually walk from the nest to the feeding compartment to get their food. So this is the whole colony was moving from the nest to the feeding compartment. This was so that observation box would become familiar to the bees. So they would show their behaviors, their natural behaviors, basically, because otherwise they would just want to get straight back to the colony straight away. So we usually let them do that. But during the test, we plugged off that observation box so that there was no bees inside it. We took a bee out and we subjected them to one of these 3 conditions. So the first one is we just didn't touch them, and then we just put them in the box and observed their behavior and recorded their behavior for 25 minutes. Second was a tactile condition, which was the bee was taken out and then we touched them on the antenna or the vertex, which is the back of the neck, for 5 seconds with a heat probe that was turned off. So it was just tactile touch. And then the noxious condition, which was when the bee was touched with the heat probe turned on, 65 degrees Celsius or 149 Fahrenheit for 5 seconds. And then the bee was put into the box and observed for 25 minutes. So here are our first results. If you just look to the dashed lines on this graph, hopefully you can see them. The dashed lines basically represent the antenna that weren't touched at all. So if the bee has the 2 antennae, some of them were just never touched because there was only 1 in each condition that was touched. So as you can see, the grooming was very low across all the time for these ones that just weren't affected or weren't dealt with at all. So that's as you'd expect. In all the conditions, this grooming was quite low. If you look to the black solid line, you can see these were the antennae that were touched with the tactile, so with the heat probe turned off. You can see grooming was a little bit higher. It wasn't so high. And then the red line, if you look to the first 2 minutes, you can see that there's a big difference between the red line and the black line. And this tells us that the bees were grooming specifically their noxiously touched antenna, so the one that was touched with the heat probe. They were grooming that for a lot longer in the first 2 minutes. And these results we expected from previous work in invertebrates about how this response, this acute grooming, should be in the first couple of minutes. So bees seem to groom the affected, noxiously stimulated antenna. And I've got a video of that as well. So hopefully you can tell it's this left antenna. I'll play it again because I think the bee shape makes a lot more sense to me, but it's like this one on this side. So it's specifically grooming this one on this left side. Okay. So then we also just looked at some other related behaviors that may have been increased. So we found that as well as this acute grooming of the antenna, overall body grooming was also increased after noxious stimulation compared to both of the groups, the tactile and the not touched. Flying was also increased, which kind of makes sense for bees because when they're in a defensive situation, it would be quickest for them to fly away from the situation. It would be like their defense response. And same with being stationary. A lot of the time, this was seen over time. So this being stationary got more over time. And this makes sense because bees would probably fly away first, and then they would panic and start to be stationary. And this backlying behavior that was also seen over time was associated with being stationary, and the backlying, they lay on their back and raise their stinger, which is obviously a defensive response for bees. So both of these were increased, and these are defensive responses that may be associated with protective behavior. So the conclusions of my research so far has been that there's mainly positive evidence for both of these indicators of pain in bees. Obviously a lot more research is needed, but yeah. And then that's it. And thank you to all of my collaborators.
Thanks very much, Tilda. We now have a 20-minute period in which you can ask questions about any of the three talks you've just heard before we then hear from Sid and Peter Godfrey-Smith. Now, please wait for the microphone to come to you so that everyone else can hear your question. And we'll start with Orion Zaks in the fourth row there. Please, please do wait for the microphone. Sorry about that. Fourth row.
Hi, thank you very much. Your talks were fascinating. I actually have a question for Andrew. So, so it's true that non-vertebrates don't have V1, but I was wondering, how would you consider evidence of some sensory processing and not others? Like, can we see this kind of differentiation? And if we can, would you consider that good evidence or still not as good as the behaviors that you're looking at?
Is this on? Potentially, yeah. I should say I'm not a neuroscientist, so Tilda's probably more the person to ask on this than I am. I think that, yeah, if we can look at the animal's brain and potentially sort of see functional analogs of the sort of human case, then that would be fairly convincing. I think the trouble with that, for example, bees, is that no one's even suggesting that there are kind of functional analogs of anything like a cortex. There's sort of Andrew Barron's argument that the sort of central complex in insects is sort of equivalent to the midbrain, and then relies on kind of Merker-esque views on what might be sufficient for sort of very basic levels of consciousness. But yeah, I don't, it's not my area, but I don't see that evidence being there. But maybe Tilda wants to add to that. Or maybe not.
I think you answered it very well. I think I would agree with the functional part.
You're underselling yourself though, Andrew. I mean, we argued in a paper called The Interplay of Neural and Behavioral Evidence that we co-authored that we need both kinds of evidence. The case can't be purely behavioral. There's a sense in which neural and behavioral work can be mutually reinforcing if done in a coordinated fashion. And so that the behavioral work can't be the entire story. Let's have a question from Matthias Michel next.
Hi, thank you. Fantastic symposium. I have a question for Tilda. So I was wondering why you think that the behaviors that you demonstrate are evidence of pain instead of complex nociception.
Do you mean all of the behaviors or just these
all of them
ones? Well, I'd like to know what you mean by complex nociception. Like,
well, I don't see any evidence supporting the view that nociception has to be purely reflex, right? You could have forms of nociception without pain, without subjective pain, that perhaps could be complex forms of nociception. So,
yeah,
you know, in order to decide what's evidence of pain or what's evidence of unconscious nociception, it seems to me that we would need some more knowledge about conditions in which they dissociate, which I don't know if we have.
No, yeah, that's kind of the point. I think we don't really have those. That once it gets past a reflex, and I think all we can say is if it gets to the brain, maybe it's becoming a little bit more complex, but like really complex nociception is kind of pain, so you can't really dissociate them at this point. So I would agree with you that we need more of a definition there. Definitely can't say it definitely is pain.
I think a lot of things can count as evidence, right? Without giving us knowledge necessarily. And I mean, you've got people who think insects don't even have brains. And then you've got people who think they do have brains, but their responses to noxious stimulation are entirely like our peripheral reflexes. And for sure, the kind of work Tilda's been doing, I think, gives them something to think about. It refutes a certain kind of strong skeptical attitude. So Dave Chalmers next.
Yeah, I guess this question is for Jonathan about the reasoning from facilitation to consciousness. You mentioned possible confounds, and there's one very natural confound, at least where phenomenal consciousness is concerned, and that's access consciousness. We know that, you know, humans, monkeys, and so on, these two things go very closely together. So where you have a possible function for phenomenal consciousness in these cases, you've also got a possible function for access consciousness. So that seems to open up the possibility that, okay, insofar as you find this facilitation in various invertebrates, it's access consciousness that's playing the role. And after all, it's already pretty plausible that there's a distinction to be drawn in, even in invertebrates, between accessible information and non-accessible information. But that seems to be wholly consistent with the idea there's no phenomenal consciousness in those systems.
I don't want to commit from the outset to the idea of phenomenal consciousness and access consciousness being distinct. I think it is a empirical possibility that they genuinely are same neural basis as certain versions of the global workspace theory would suggest. I also don't want to commit to them being identical at the start of inquiry either. I want to try to remain neutral on that. The approach I like most of these issues is Nick Shea's approach in his paper called Methodological Encounters of the, what is it? It's a wonderful, witty title that is impossible to remember and has no search terms in. So it's a sadly neglected paper, Methodological Encounters with the Phenomenal Kind, where, I mean, this is Nick's view as well, right? That we open-minded at the start of inquiry about whether phenomenal and access consciousness are identical or distinct, but we commit to either way, they will have functional markers that we can study the clustering patterns of. And one possible outcome of that inquiry is that we find that not all of the markers of phenomenal consciousness run through access, and then we'd have grounds to think them genuinely distinct.
Do we now have any grounds to think that those patterns of facilitation are more closely tied to phenomenal consciousness than to access consciousness?
Well, so at present, I think we're at an early stage in inquiry where the right thing to do is to be open-minded about whether we're looking for two distinct kinds or a single kind. Yeah, there's another question from the front row. Let's take that one.
So I have a question for you about trace consciousness, trace conditioning versus delay conditioning, which is clearly what you have in one case is working memory and the other case you don't. And that's even what your experiment showed.
Yeah.
By the way, I applaud. This is a fascinating symposium, and I applaud your approach. But I don't see any prima facie reason to believe that the difference between that consciousness is what makes the difference.
Yeah.
I mean, just the very presumption. Why do you think there is?
I think so. What I want is to systematically inquire into whether there is. So I agree that at this stage of the inquiry, one could hold the view that most probably working memory is distinct from consciousness. These differences in the flies and the bees are driven by working memory, not by consciousness. And so one might bet that the end result will be insects turn out to be a wonderful model of working memory and not of consciousness. But I think it's very soon to be prejudging that kind of thing. And let's be open-minded. In our human and mammal work, let's try and look as hard as we can for differences that are associated with consciousness and that can't be reproduced by unconscious working memory. And then let's go and look and see if we find those in a wide range of other animals, being open-minded about the idea that we might find the conscious-unconscious contrast in those cases too.
I agree with that.
All right, so sorry to stick to the front row. We'll have Ned Block next.
One question I have is also for you is, and I also agree with Susan, this has been a great symposium. What reason do you have to think it is working memory rather than iconic memory? So, the one difference is that iconic memory is much more easily disturbed by various kinds of masks whereas some kinds of masks will work, so different kinds of masks could be used to distinguish. So that would be really interesting. I would be more impressed if it was iconic memory. So then the difference because iconic memory, you can make at least some probability of an argument that can't be unconscious.
Yeah, no, I mean, it's a great question. I think my read on this, the Drosophila study was that the sensitivity to distraction seems to point to some involvement for working memory. My thinking there was that this is a kind of memory that is gated by attention. In some sense, attention controls the gates. When you administer an air puff, it distracts, something else gets in and it stops the trace conditioning. So there's perhaps some tentative evidence to think this is working memory, but it's one study, and let's absolutely keep open-minded about the idea that there might be forms of iconic memory at work in insects as well. Let's go further back now. Let's go back to the fourth row, and then we will try and work our way to the back of the room. Thank you.
Can you hear me?
Yep.
Thank you. The answer to this question, if animals have consciousness, is yes.
Mhm.
Yeah. And my understanding, sure, I'm a little bit nervous because it's my first time I'm in this confidence of consciousness and first time I'm speaking. So my understanding of this problem emerges from my theory, how physical universe gives rise to consciousness. This theory is overwhelmingly supported by theoretical and empirical data. And my theory recommended for testing by Ken Wang and Jonathan Fritz from National Science Foundation for testing.
Yep. I mean, I think that's, we get the question, there's many people in the room who have strong theoretical commitments, and they really have a theory of consciousness that they believe is very strongly supported by evidence. And I'm here saying, well, let's not premise research into animal consciousness on any particular theory. In the end, I think that's because we've seen during this meeting an enormous amount of disagreement as well, suggesting it would be a huge risk to commit at this stage to any specific theory.
I know, I realize that.
I mean, I think we're in agreement on that in a little short of time. Is that all right?
I will very quickly. So my theory accounts for subjective experience, meaning, and verbal report. It explains how neural events are transformed in subjective inner experience. Answers the question, what consciousness is.
Yeah, I mean, I think that's fair enough. Lots of people in the room have their own theories and would say exactly the same thing about their theories. Is it all right? We move on to the next question, which I'd like to take from Henry Shevlin. Sorry to cut the question off a little bit there. Henry?
Thanks. Fantastic data. Just a question for all of the panel, really. It's obviously becoming increasingly common. We talk about animal consciousness, invertebrate consciousness, insect consciousness, but those are still quite large groups. So I'm curious if you get any sense as to how much sort of predictive validity work on Hymenoptera has for other insects. I mean, it could be, for example, Hymenoptera, the superstars of the insect world. I just wondered if you've had any insights at this stage on how much these kind of risk results tell us.
I was talking about Drosophila in one of those studies there. Would Matilda and Andrew, would you like to come in on this?
I was just going to bring up Drosophila as well.
Yeah, we've got at least two extensively studied groups.
Yeah, and I think it's like the more that they're studied, the more we have positive indicators. But those are just the two most studied. So that would. Do you want to?
Yeah, I mean, very fair point. Like there's 60,000 vertebrate species versus well over 1 million insects. Obviously, work on the buff-tailed bumblebee is not informative on all of those species in the same way that work on lab macaques is not that informative on black-tip reef sharks and much, much less related species in insects. And I think as well, even within Hymenoptera, there are about 100,000 described species, possibly more like sort of 400, 500,000 undescribed ones. Most of them are parasitoids. I think it's very reasonable to imagine that parasitoids probably might lose they do lose an awful lot of kind of cognition given their lifestyle. So can we generalize? Biologically, no, but given the lack of any data on most of these groups, I think sort of have to for the time being. And so, if other people do want to study understudied insects, that's fantastic.
Well,
for now,
we do ourselves, right?
Well,
there's some very commercially important insects like black soldier flies that have not been studied at all, and this is a big problem that we want to remedy. So Henry, there's a question from 3 rows behind you then.
Thank you. I was wondering whether you are thinking about how we can avoid the anthropocentrism, trying to project the way we think consciousness functions in humans, how it works for us, whether we can find ways to avoid that. For example, I don't know, with some training of a network identifying patterns of behavior that we haven't thought of, maybe could be a way. I don't know, but I was wondering whether you are thinking about it.
Thoughts on avoiding anthropomorphism?
Yeah, I think up until really quite recently in animal cognition work, that was pretty much the worst criticism you could level at someone, that they were being anthropomorphic. I think increasingly in the last couple of decades or so, there's been a realization that you can go too far the other way. Frans de Waal, I think, calls it anthropo-denial, where there must be some point at which, you shouldn't be assuming too much that animals are human-like, but you can also go too far the other way, where there must be a point at somewhere, we don't know where necessarily that is, but there must be a point at which you're eliminating evidence from humans too much. And so I think, given that humans are the one kind of absolutely the case where we absolutely know for certain that yes, this one species definitely is conscious. Maybe some philosophers will disagree with that. This one species definitely is conscious. We do need to take, I think, that as a paradigm case. But some people argue for more bottom-up case. I
also think a huge amount of work in consciousness science is premised on other primates in particular also being conscious. And so let's not deny that, we are already studying animal consciousness. Let's broaden the range of animals we're studying. There's time for a couple more questions. So there's one here from about 8 rows back in the middle, making the really helpful microphone people do a lot of exercise here. Thanks.
Thank you so much. It's terrific, terrific research. And I'm going to have to think about insects now. I was coming around to fish, and now, insects. And I want to push you on flexibility, because that seems to me to be the really interesting place where, in that cluster view, we move from the sort of cluster of things that does require conscious, processing. And so, so grooming behavior is interesting. But it looks like that is behavior that could just be, a kind of instinctual, responsive defensive mechanism. So I like the trace conditioning where you get distraction. I think that research is really, really interesting, and with the fish, Victoria Braithwaite did a novel object, and fish with the novel object, when they were in pain, would, not avoid it. They wouldn't attend to it. And that seemed to also be a really interesting additional sort of evidence for flexibility. And I wonder why you're calling grooming behavior flexible in that way, or how you guys would even, I'll push you even further, how among the three of you, you would define what flexibility is.
Defining flexibility. I mean, I can answer the grooming one, but maybe I'll give that one to the others. But yeah, I think you're right. Like the acute grooming, especially because it's acute and short term, could definitely just be an instinctive, reflexive response. I think the point is more that we do that, and in humans, it reduces our experience of pain. Like grooming and rubbing the injury straight afterwards, it does reduce our experience of pain. So it's sort of associated with that. And if we assume the same thing, then it's definitely reducing some sort of noxious processing in the insects, and they're actively trying to reduce it. So whether they're feeling pain at the same time as that happening, yeah, you can't tell. And whether that's a reflex or not, we can't really tell at this stage either, but it's a good point.
I would add that in my own case, what I find quite evidentially relevant is evidence that points to the response to injury being under centralized rather than local control. So, for example, in some of the octopus work with Robin Crook's study of octopuses, you apply acetic acid to one of the octopus's arms. It will use other arms to wrap around it and nurse the site. It will also scrape away at the stimulus with its beak. And so that information is sweeping around the entire organism, and there's centralized coordinated response. And to me, that really is relevant evidence. So we have probably the last question now. Let's have Albert Nguyen. Second row. Thanks very much to those helping with the microphone.
Yeah, thank you very much for the very nice presentations. I'm wondering whether you may have to characterize whether all of you are open-minded, that you may not committed to what most philosophers presuppose, that there's one and only phenomenal consciousness, but especially to the different variants of investigations, although they are sometimes, most of them are driven by human psychological investigations, they may nevertheless indicators of a rather different quality of phenomenal consciousness that is realized in different species.
Yeah, I think when you think you've got good evidence to infer that another animal is conscious, it's just the beginning, because you then immediately face the question of, well, how does the structure and content of its experience vary with respect to our own and with respect to other animals. And I've been thinking about that too. We have the Dimensions of Animal Consciousness paper that Albert has written a follow-up to. I think it's another part of this research program that let's make sure we don't stop at the question, is it conscious or not? But let us also ask, what is the structure of its experiences along relevant dimensions? Tilda or Andrew, any closing comments on this?
I think we've got a...
Yeah, I think so. We've come to that point where we all want to hear from Peter Godfrey Smith. So let us all welcome Peter to the stage for his talk. Bring it back down.
Okay, this is the forwarder. Yes. Okay, it's great to be here. I'm also giving a talk at an NIH conference in a few days that will be streamed, organized by BU He, that is a continuation in a way of this talk. The two will overlap, but the second one will add some more animals. What I'm going to do is handle things by making use of a positive view of the biology of felt experience or consciousness in a very broad sense. So it's a bit the opposite of Jonathan's theory light approach. Now, I can't in a talk this short defend that view very much, but I can indicate how some lines of inference work, how I get to it, where it takes us. So I'll start with that, and then I'll look at some work on animals. Okay. This view I have introduced sort of in pieces in some earlier work, but I'm going to sort of make it a bit more systematic and christen it today. In a rough way, we might distinguish three kinds of features used in general accounts of the biology of consciousness. Firstly, what might be thought of as broadly agential features, how the animal handles itself in the world. Secondly, information processing features that have a tie to brain architectures, the cortex, things like that, though you might be able to set up functional analogues of these. And thirdly, more lower level or basic features of nervous system activity. Now, this is my first ASSC, but I think we could say that a lot of work here is sort of oriented towards the second of these. It distrusts the first because we've learned that much coherent processing can be unconscious, and it tends to regard three as just mechanistic detail. My view, in contrast, is based more on 1 and 3 with less of 2. Now, I can't yet say this is a definite model. It's more an approach, a bet about the shape of the truth. It has two elements. It starts by embracing an idea that sometimes, for example, by Tom Nagel yesterday, used to formulate the mind-body challenge, the idea of subjectivity, the first-person point of view. Subjective experience is the experience of a subject. Where does that come from? Well, a specialty of animal life is action, agency. With agency of a sophisticated kind comes subjectivity, acting on the basis of how things seem, having a point of view, making valuations. The other part of the view is the idea that nervous systems have a special combination of features that figure in bridging the explanatory gap between mental and physical. Nervous systems have a pair of properties. There are the networked, relatively discrete cell-to-cell properties that enable computation, and there are what I will collect together as large-scale dynamic properties. Here I have in mind the more diffuse, often oscillatory dynamic patterns that are picked up in an EEG and similar technologies, the properties that involve more the global electrical activity of a brain. Now, I think that felt experience, consciousness in a broad sense, is characteristic of systems that have this combination of properties together. They are configured as subjects with a relationship to their milieu of a kind that we roughly characterize with terms like point of view, and they achieve this with neural activity. Nervous systems are control systems of a special kind. They're inherently active with large-scale dynamic patterns where that activity is modulated by sensory events and other events in concert. Why do I think those features of nervous systems are special? Well, I do appeal to empirical arguments made by many others, Varela, Singer, Maloney, Van Swinderen, some people are cited there. This work on the cognitive and experiential role of brain oscillations in particular is of intense interest to people in this group, and I approach it as a non-expert with some caution. But large-scale dynamic properties appear to play a role in integration, in selective attention, sleep-wake cycles, anesthesia, and so on. I also want to make a mapping between the view of nervous system activity I'm employing and some phenomenological features of experience itself. I think that what is basic to ordinary human experience is what I will call experiential profiles. I resist what John Searle has referred to as the building block view of conscious experience, where there's this item, then that item, perhaps combined together. An experiential profile is a total way things feel to someone at a moment. These profiles are inherently multifaceted, gestalt-like. There's something or some things in attention, other things in the background. These may be gist-like within a particular modality. There's also bodily awareness, and the whole is modulated by things like mood and energy level, which are usually far from attention, but part of the totality of what it's like at a moment. In Ledoux's talk on Thursday, he discussed what, after Tulving, he called anoetic consciousness. And he said this includes things like a feeling of rightness. He called these fringe states. I think of these as more subtle parts of experiential profiles. Now, I take this feature of ordinary human experience as real, non-illusory, and I conjecture that it is characteristic of experience in general, not just in humans. This is part of what felt experience is. From here, I think back to that picture of nervous system activity. As has been noted by a number of writers, Pisano, way back, Ginsberg and Jablonka, others, when neural system has the form of large-scale patterns with modulation, a range of different senses can have the capacity to affect any given state. An internal state strongly affected by what the animal is, for example, seeing, will, if the animal is mobile, also be affected by ongoing tactile sensing, proprioception, energy level, and other things. So I think the gestalt-like character of experiential profiles, with the ebb and flow of salient modalities and attention and so on, is naturally explained by this view of nervous system activity. The nervous system features I'm talking about here are not just how control happens to be done in us. I think they give human behavioral control some subtle functional features, which have to do with repeatability, timing, integration, and so on. So that's my view. This has some relationship to what Ned Block calls a biological approach to consciousness in its contrast with some other views in this area. And I think it's worth now having a label for this position. I'll refer to it as the NDS view, where that stands for neural dynamics of subjectivity. Now, more details and more of the motivation can be introduced by looking at the evolutionary side. Nervous systems evolved perhaps something like 600 million years ago, around the time of the White Sea Ediacaran, animals like this, or before. The question of single or multiple origins is unclear because of comb jellies, but nervous systems are found in nearly all animals. Their evolution is so early that it was before what we usually think of as the characteristic regime of animal life, with movement, interaction, predation, and so on. Very early nervous systems may have been largely concerned with just pulling the animal together, taking the active capacities of our protist ancestors and making coherent action possible on a larger multicellular scale. Nervous systems in general, so there's that Iacoran slide, nervous systems in general, as far as is known, have an oscillatory tendency. Back in the 1960s, the neuroscientist L.M. Pisano and others explored active large-scale oscillations in hydra, a very small jellyfish-like animal, a freshwater hydrozoan. Pisano himself conjectured that patterns like these in an early animal are ancestral to brainwaves, EEG-type patterns, and the like in more complex nervous systems. After all this, we reach the Cambrian from about 540 million years or so ago. Now we encounter a regime of interaction, predation, with sensing including good vision, hard body parts in animals, movement and manipulation. This coevolutionary regime led to a firming or a focusing of subjectivity in the service of real-time action. So all those point of view related properties and probably rich kinds of evaluation and learning come on board, or more strongly on board here. Marked conspicuous behavioral complexity evolved in several different lines. Arthropods, who were first, vertebrates like us, and cephalopods, a group of mollusks, an unlikely phylum for this. The branching events that separate these groups, crucially, took place before the Cambrian. They took place in the quieter time. When behavioral complexity evolved, it evolved separately in these three groups with different bodily resources and lifestyles and very different brain architectures. Now, Nick Strausfeld, Gabrielle Wolf, and their collaborators have argued for hidden mappings of design between arthropod and vertebrate brains, but those would be very deep and subtle, and cephalopods are not even included in this claim. Even if what Strausfeld refers to as a basic executive brain evolved very early and links arthropods and vertebrates, mollusks threw this design away, and when cephalopods became behaviorally complex, they did it all differently. So nervous system architectures and body plans differ. Basic features of nervous systems, including that combination of network features and large-scale dynamics, are held in common, are conserved. The project of generating coherent agency and matching actions to the state of the world is also common across these groups. In each case, we get acute senses, high-resolution image-forming eyes, instrumental learning, navigation, and so on. So these animals share some of the 1 and 3 properties and much less of the 2. Convergence on integrated agency, conservation of basic features of nervous systems, and very different brain architectures. Some distinctive neural features that seem to matter to experience in us may have a similar role in some of them. Bruno van Swinderen's lab at the University of Queensland studies large-scale neural oscillations in flies, Drosophila. Earlier work from that group showed an association, some association between oscillatory activity and attention in these animals. A newer paper from 2020 with Martina Grabowska as first author shows that attentive locking onto particular objects in the visual field is associated with beta oscillations, 20 to 30 hertz in the fly brain. So objects were created in the fly's visual field, a similar VR system to one that was talked about earlier, with different flicker rates and other differences such as size, which flies care about. And when flies attended to one object or another, their beta oscillations became phase locked to that object's temporal features. Attention could also be redirected between objects by stimulating a reward circuit. So a normally less appealing object within the visual field could be made more so. That stimulation was done with optogenetic stimulation of a sort of reward arousal mechanism. This paper claims that beta oscillations are used to regulate selective attention to particular objects in flies. Now, with these views about oscillations, there's always a question of whether they are byproducts rather than causally active themselves. And there's material in the paper bearing on this, but suppose the oscillations are not a mechanism of selective attention, but are just a byproduct of it. We are still learning about the existence of selective attention of quite a subtle kind that can be modulated by reward in a fly. Okay, I will now turn to octopuses who have their own special, contribution to make here. Octopuses have larger nervous systems than any other invertebrates, except perhaps their cuttlefish relatives. They have rather vertebrate-like oscillatory patterns. They also have a behavioral feature that is telling in the context of skeptical views about invertebrate consciousness. Now, I was going to cover the pain material that Jonathan mentioned briefly in question time, but if I'm down to 2, I'm not going to be able to do that. So I'll just talk about a more anecdotal, but I think still telling feature of octopuses. If you ask about immediate behavioral markers of consciousness within views that emphasize how much behavior can be unconscious, such as Dehaene's view, a reply might be, well, novel behaviors that engage with novel objects in non-routine ways. Octopuses of some species have an inquisitive exploratory way of being. At the Octopolis site in Australia, in Booderee National Park, octopuses encounter cameras lying around by us. They are often very interested in these objects, take them home, try to take them apart, and so on. So if a person was to say their large vertebrate range nervous systems just have the wrong layout for consciousness, this would be the claim that an exploratory, apparently highly attentive engagement with the world could be wholly unconscious. Maybe that's true, but it's not the usual sort of thing associated with unconscious processing. Okay, I'm going to have to skip octopus pain for time, and I'll give a quick summary. Okay. So in our case, we find the 3 features: sophisticated agency, a brain architecture, nervous system with various properties. We look at the evolutionary situation and see deep branchings before the origin of behavioral complexity, separate evolutionary lines that evolve that complexity on different body plans. All these animals can navigate, learn, attend, see well, and so on. They also have the shared resource of a nervous system with that special combination of properties, networked and more diffuse integrative. They use that to achieve agency. You might say none of these animals has the right sort of brain. Octopuses make that difficult by their display of consciousness-related behaviors, even by the standards of rather exacting theories. And the alternative here is not that they have different experience from us, which they surely do, but none at all. So suppose they are conscious, the octopuses, conscious in a broad sense. They are in despite their weird architecture. This frees us up to take the marks of felt experience seriously in other organisms with the wrong architecture, such as flies. There's, on this view, massive neural multiple realizability, not the same thing as total multiple realizability of the sort that readily includes artifacts, but massive neural multiple realizability. That's all I can do today. And as I say, I'll take these themes further at the talk at NIH, which is being streamed.
Thanks, Peter. We will have time in the Q&A and you can tell us about octopus pain. So I'm also going to talk about pain. I have nothing to disclose, except I thank the ASSC for inviting us and sponsoring this program. So in non-paradigmatic humans with brain injuries and disorders of consciousness, we're wrong about 40% of the time when we try to diagnose whether or not an individual is conscious. And that's using a clinical diagnosis that's based on inferences from observable behaviors that would, in typically functioning conscious humans with typically functioning brains, indicate conscious awareness. I think we should correspondingly assume that we're wrong at least 40% of the time when it comes to detecting consciousness in non-humans who do not behave like paradigmatic humans. I suspect we're wrong a lot more than that. For reasons that my colleagues have already discussed today, it can be especially challenging to conclusively detect consciousness in non-mammal animals. So my plan today is to discuss some ethical questions about potentially harmful consciousness research on non-mammal animals, the link between consciousness and welfare, and specifically focusing on fish pain and welfare, and whether these concepts of inductive risk or equipoise have anything useful to contribute to the question of justifying this kind of research. And I'll end with some unresolved questions. So across moral theories, it's pretty typical for it to matter whether or not the being under consideration for moral status is conscious. Consciousness is viewed as a necessary or sufficient condition for moral status, or at least an important trait of entities that matter morally, on the assumption that consciousness is necessary for an entity to have interests that matter. I'm going to assume this is right, and also assume that causing harm to creatures who can suffer is prima facie wrong and requires some justification. I don't actually think that justification is met in the case of using a lot of animals in research or killing them for food or for sport. In practice, lots of harms to lots of animals are accepted as morally permissible and are justified by human exceptionalism, typically. For conscious animals, that requires, if we care about consistency, sort of decoupling consciousness and moral status or the wrongness of harming and introducing some other valued trait or characteristic or capability in addition to consciousness, species membership, rationality, and so on. There are no traits or capacities that I think are uniquely human, which is one of the reasons I think that the common arguments for using animals in harmful ways tend to fail. But for welfare-related policies, guidelines, and regulations, consciousness matters, and minimally at least, by making a creature a subject that has welfare. So broadly speaking, there's sort of two types of consciousness research using animals, and one type uses animals that we know are conscious, mammals like monkeys, mice, cats. A lot of this research can be very invasive and harmful. One aim, though, is to learn more about human consciousness. And there can be a clear ethical dilemma with that kind of research if we accept that harming conscious creatures is or can be morally wrong. The other type is what I'm going to focus on today, which is research on animals not known to be conscious. And one reason to do that research is to determine if that species or taxa is conscious and or sentient. Whether or not that animal is conscious can bear on whether or not doing that research, when it's harmful, might present us with an ethical dilemma. So a few examples of research methods: mirror self-recognition tests, which are controversial, evidence of self-awareness. A growing list of animals have passed this test: chimpanzees, cleaner wrasses, dolphins, elephants, orangutans, magpies, horses, crows, and ghost crabs, I learned recently. Controversy about whether this test and what it tests for exemplifies the theoretical and methodological uncertainties of looking for consciousness in diverse species. We can also use methods like non-invasive neuroimaging, like fMRI, invasive brain implants, brain lesioning, noxious stimulation for pain processing. There are a lot of potential harms, including harms that are specific to captivity in some cases, like deprivation or isolation. We have behavioral evidence. We've talked about some of this today, of consciousness and sentience in many animals, including humans, from which we infer consciousness. Verbal reports are a type of behavior. Relevant animal behaviors are not always well understood, even in mammals. Not all scientists who study consciousness are also scientists who study or understand animal behaviors. In nonverbal humans, we often rely on behavioral evidence, although the meaning of behaviors is not always clear. Human babies cry a lot. Why are they crying? Are they in pain? Are they hungry? Are they sleepy? Right? We also have diverse neural structures owing to this evolution of brains and consciousness multiple times across multiple taxa. So birds lack a neocortex, which is thought by some to be essential to human consciousness. But I don't think there's a lot of debate about bird consciousness. I could be wrong there. All right. Oops, I mixed up my pages. There we go. So the existence of conscious pain in fishes is controversial. Whether there's genuine controversy depends on who you ask, but one type of experiment on fish pain induces a noxious stimulus, like injecting acetic acid into the fish's lips. And the fish will breathe faster and rub the affected site, which are typical pain behaviors. And they also won't engage in sort of normal anti-predator behaviors when they are in pain, but they respond to analgesia like aspirin or morphine. They have opioid receptors. So from this, via inference to the best explanation, we can infer that fishes feel pain. And fishes also engage in lots of interesting and complex behaviors, including some that involve memory, social learning, recognition of others, cooperation. The cleaner wrasse has passed the mirror self-recognition test. The arguments against the existence of conscious pain in fishes mostly focus on fishes lacking the human-like neural architecture for pain, which is a deductive inference. One skeptic also claims that fishes don't behave as if they experience pain, which is an inference to the best explanation. Over lunch, we were discussing whether sharks do behave as if they experience pain, and there's still some question about that, but teleost fishes, the finned fishes do. So Bowley et al. Performed an experiment involving noxious stimulation, an electrical stimulation of the median nerve, which apparently is very unpleasant. And the subjects were humans. They were minimally conscious patients and patients in the vegetative state unresponsive wakefulness syndrome. They used PET scans, which revealed similar activations in the so-called pain matrix in MCS patients and healthy controls, but not in the VS-UWS patients. And they concluded from this that MCS patients experience sensory and affective perception of pain. This was an inference to the best explanation. The aim of this research was to determine whether MCS or VS-UWS patients experience conscious pain, and if so, to try to limit or treat that pain, especially procedural pains that they might be subject to. This is also a potential objective non-behavioral test to distinguish between these two groups of patients who can behave in very similar ways. So the experiment could actually potentially benefit not only other patients, but the research subjects themselves. But we have no similar benefits to fishes from being experimented on to see if they experience conscious pain. So as I said, I don't think the standard justifications for using animals in research typically hold up under very much scrutiny, but I want to look at a couple of other possibilities for justifying consciousness research in animals who are not known to be conscious or sentient. So the concept of inductive risk is useful for thinking about research with animals. The idea in a nutshell is that there are interacting epistemic and ethical risks whenever we accept a scientific hypothesis. So if our experimental hypothesis here is fishes do not feel pain, the epistemic risk is just the risk that we're wrong. The ethical risks are going to be the ethically significant consequences of being wrong. So in our experiment, if we're wrong, the fishes will experience pain and be harmed. So Rudner, back in the 1950s, provided what I call the principle of inductive risk, which is that our epistemic risk taking should be constrained by the ethical risks of being wrong. So high ethical risks should limit the acceptable epistemic risks. So we have existing evidence of diverse Non-homologous nervous systems across species and taxa, plus pain behaviors in teleost fishes, plus complex non-reflexive behaviors in fishes. But fish lack a neocortex, and their brains are not like our paradigm brain, the mammal brain. If we're wrong that fishes do not experience pain, then our experimental fishes will suffer. Is that enough to constrain epistemic risk-taking? I think that it is. The existing evidence is strong enough to constrain epistemic risk-taking and limit consciousness research that is potentially harmful and painful. And another way to say that is that the existing evidence increases the epistemic risk of the hypothesis that fishes do not feel pain. So one thing you'll notice about inductive risk is that it indexes epistemic risk taking to ethical risks, but it doesn't consider whether or not the research in question is beneficial to anyone. So it would permit low ethical and low epistemic risk research that is, for example, purely curiosity driven, where the main benefits might be epistemic, but it wouldn't permit high risk research even if there were high epistemic benefits. Another concept, and this one is not typically used in the context of animal research, but is used for clinical research with human subjects, is equipoise. Equipoise is useful when you're designing a randomized clinical trial with a new medical intervention, right? It requires that there's no superior intervention already in existence. So this state of equipoise or true uncertainty exists when there's no good basis for choosing between two or more options. This is especially important when the experiment's potentially harmful or when an important benefit, like an effective treatment, might be denied to the subjects. So this would permit potentially harmful research if that research has the potential to break equipoise and resolve the uncertainty in a way that's potentially beneficial. So Bowley's pain research would have been permitted here, despite the risk of causing suffering, because there was genuine uncertainty about pain perception in those two categories of patients. So if there's genuine uncertainty about conscious pain perception in fishes and a potentially painful experiment could resolve that uncertainty, it would be permissible to conduct that research if breaking equipoise is potentially beneficial. So equipoise again requires a benefit. I'm skeptical that there are substantial benefits to fishes of harmful consciousness research. Will it change research regulations to include fishes and fish welfare? Will fishing and aquaculture practices be subject to regulation if they have to consider fish welfare? We still permit a lot of suffering and distress in mammals, including in research and in food production, despite having really no reasonable doubts about their sentience. If breaking equipoise could be beneficial for fishes, all things considered, it would permit research that causes fish pain for the sake of resolving the uncertainty about fish pain, assuming that uncertainty remains genuine. Inductive risk, though, would call on us to limit those ethical risks, and it would also require that we really adequately understand the epistemic and the ethical risks. So there are a lot of unanswered questions about non-mammal consciousness and sentience, and I think answering those questions, I think it ought to come before we engage in potentially harmful research. There are still lots and lots of questions about human consciousness and the consciousness of other animals, including questions about its ethical significance, the ethical significance of consciousness. We ought to know what would count as robust evidence of consciousness and sentience, things we've discussed here today, before we go poking around for it. But we also ought to have a genuine commitment to accepting and acting on that evidence if we find it. In the case of non-mammals in particular, there are still a lot of unanswered questions about the implications for their welfare and about what counts as good welfare. So what's a good flourishing life for a fish or a bee or an octopus? Thank you. So we have 18 minutes left.
Other questions?
Sure, you can do that. You did a great job.
It's our final Q&A period now. So questions, especially relevant if they're for Peter or Sid. Let's please wait for the microphone to come to you. I'm not seeing questions. There must be questions on the incredible claims from Peter's talk. Let's start the second row. Thanks.
Yeah, really interesting session. I'm thinking that you are dealing with systems that are quite different from us vertebrates in this room. Is your project in any way relevant for detecting consciousness in AI systems, or is that a completely distinct issue?
Any thoughts from Peter or Sid first?
The view I was describing has consequences for that, certainly. If we look at us and we think we've got this cluster of features that seem important, look at other animals, the architectural stuff is very different. The neural stuff is preserved, including what look like experience relevant features of the neural stuff in us. It's preserved. And from then I infer, well, maybe the neural stuff really does matter a great deal. If that inference is a good inference, then if you build an artificial system that mimics some of our architecture but doesn't have the biological properties, you've probably not built something that is a sentient or conscious system. So that would be the straightforward implication of the view I was describing. Now, then you might think, okay, I'm going to build a different sort of artificial system that might not be alive, but has many more brain-like properties, including the ones that are so conserved and apparently so important in animals. And then you'd have a more difficult problem of adjudication. But I think in the case of systems that are built on a conventional computer hardware and just are supposed to have some broad structural features similar to ours, you know, I'm quite skeptical about them being conscious.
I think I have a sort of quite strikingly different view from you on this, in that I am extremely open to the possibility of consciousness in AI. I think it's very unclear to me whether these neuron level properties you're describing matter, but also even if they do matter, it's not clear to me they can't be emulated in AI. So I wrote a piece recently in Eon, the headline of which was to test for sentience in AI, first understand it in animals. My hope is that this research program in animals will result in us having better criteria.
When you say AI, do you mean using an ordinary computer hardware?
Yeah, I mean, stuff along the lines of what already exists.
Right.
So I want to correct my bias towards the front of the room if possible. So questions from the back of the room. Let's
Actually,
have one from
I wanted to say something about that.
Please do, yeah.
This is apparently something I'm talking about at NIH as well in a couple of days. I think one of the issues with AI is that our current reliance on behavioral evidence is really problematic there, because robots in general right now already can mimic animal and human behavior, and they can certainly mimic verbal report. I mean, we've already had people convinced that some of the chatbots are sentient, right? I mean, they can say, you hurt my feelings, right? Or I'm in love with you. And so the question then is, you know, a question of credence and what we do with those kinds of behaviors. And if we don't believe them at all, is that the right approach, or do we need to give them some credence?
Back of the room.
Okay, so thanks a lot for this very interesting symposium. I had a question about painkillers. So what they really show when animals, whether it's a fish, or I've seen that there was also information, something about with bumblebees, like taking the equivalent of morphine. When we take morphine in humans, it's okay, it's to relieve from the pain. But is there the only possible explanation? And is there not, like, it's a naive question, like biological effects that are just about, I don't know whether it's dopamine or anything else, that could explain the use of these substances independently of any feeling of pain?
Should we start with Sid?
So in fish, so in animals with an opioid receptor, right, which humans also have, the suggestion is, and I'm not a scientist, I'm an ethicist. The suggestion is that if they are responding in a way that indicates that they no longer feel pain after being administered an opioid, that's evidence that there was pain to begin with, right? I vaguely recall, though, that opioids don't actually sort of kill pain. They're not analgesic. They have a different effect, right? In humans, at least. So I don't know that I can adequately answer your question. Maybe someone else.
Could I might sketch the crook octopus pain paper just quickly, because it includes a painkiller element. This might be helpful given it's come up a few times. As Jonathan said, acetic acid injection in one arm. And what was important about the study was the network of different consequences this had. There was a switch in preference for a place. A formerly preferred area was no longer preferred because something bad had happened there. There was a second condition place preference, which relates to the analgesic. So the octopus, in some cases, were given an analgesic chemical, and that was associated with a particular normally not preferred chamber in the environment. That then became the preferred chamber. And there was also grooming of the injured spot, and there was some physiological evidence as well. But just responding to your question about the painkiller, the fact that the painkiller switched a preference for a place, what would be the non-experiential sketch of a reason why that would happen? I mean, I get the idea that there could be other pathways, as you say, but is there a particular one that you have in mind?
Think of the study by people like Berridge about people with addiction and the dissociation between wanting and liking, and that in addiction, you can have, they no longer have pleasure out of the drug and they still want it. So it's not that there is necessarily a hedonic tone in this kind of behavior. And I was wondering whether, so this is for the pleasure side, but whether we could have the equivalent for the negative side, and the disliking and diswanting could be also dissociated. But we can.
Never thought of that. That's interesting. Yeah, that never thought of the flip side of that. Yeah,
that's nice. Yeah, let's have a question from 5 rows down from the back, in the middle of the row. Yep, you. Please wait for the microphone to come to you.
Yeah, thanks. Great panel. So there was a ton of consciousness research on non-human animals in the 19th century, and a lot of it was extraordinarily brutal. You know, they're cutting out just about any brain structure you can think of, brainstem. It's really brutal. I guess I have 2 questions. I guess this is mainly directed at Sid. One is, I don't know, is there like a wicked data problem in non-human animal consciousness research now? Like, it's probably hard to disambiguate what we know about neurophysiology in non-human animals. That's sort of comes from extremely brutal research. So that's one question. And relatedly, are there specific techniques that are being used today that you would sort of want to see put an end to? Like, are there specific ones that you have concerns about?
So I think this is one of the reasons why thinking about this in terms of risk, in terms of both epistemic and ethical risks, is really useful, because it allows us to accumulate evidence. It doesn't say you cannot accumulate that evidence, but once you have, right, it has to be considered on the risk side when you are when you have a hypothesis that is not consistent with the existing evidence. And I think that you're right. We have a lot of evidence that perhaps we're not taking into consideration, or that we're unduly skeptical about. The questions about fish pain are those kinds of questions, right? There, we have all this other evidence about, you know, fishes experiencing pain, but they don't have a neocortex, right? They don't have this other thing that humans have. but we have reasons to think that there are creatures other than humans who do experience pains. Mammals, certainly, and most of us are familiar with that, but the more distant animals, the octopuses, the fishes, the insects, we may still have legitimate reasons for doubting in some of those cases, but I think that once and I don't know how to describe what the point is where the evidence, flips the risk from, overwhelmingly unrisky to overwhelmingly risky, but there is some point where that happens.
Yeah, I mean, I want to see serious discussion of the harms and benefits of the research that is being done in this area, as a normal part of these conferences and a normal part of the talks we give, the conversations we have. Anyone else like to comment on this, or should we take another question?
Okay.
Yeah, we'll have another question. These people at the front are so keen. I have to go to them. So third row. Please wait for the microphone to come to you. The questions from the back are also welcome, of course.
Since Kent Berridge's work was mentioned, the opioids go on the liking, not the wanting side of his dichotomy. And it's not a mystery why you should have place preference for a place where you got opioids, since opioids are they don't just go against the aversive component of pain, not the sensory component, but the aversive component. They also can code hedonics in some parts of the brain, including in some of Berridge's hotspots. So opioids do different things, different parts of the brain. You may have an opioid producing positive hedonics, liking or pleasure, and in a certain place, in a hotspot in the ventral pallidum, and a little distance off, it might be aversive. But overall, generally, it is the sort of thing you would expect an animal to like, and to give it place preference on the whole, although with a pipette putting in different places, sometimes positive, sometimes negative. So I think not that mysterious. That's something that can block the aversiveness of pain and also can code hedonics, should have those effects mentioned.
A word on that. Right, that raises a question of, I can't remember whether Crook, as part of her controls, included a situation where the painkiller was given, but the aversive injection of the acetic acid was not given. And it raises the question of what is the role of a control like that, given that that the painkiller could be doing both. It could be genuinely helping with the painful stimulus, but also quite pleasant even if you didn't have the painful stimulus. From memory, I don't know if she did that, but I could be wrong about that. Can I just use this also to segue to a comment that follows? I've forgotten the gentleman in white's name in the front who asked a question in the first round, but we had a bit of an exchange like this at the John Nicodes, as I recall. And I think I've thought of a better way of replying to some of the things that you say.
A year later.
Yeah, because it bears directly on this. What's important about the Cook experiment is that the contrast is no longer between well, here are three things. You've got a sort of integrated, multifaceted response to an aversive event with experience, an integrated multifaceted response to an aversive event, in scare quotes now, without experience, and you've got more peripheral, less integrated, less complex responses, such as reflex withdrawals and things like that. And the Crook experiment at least takes us to a place where we're no longer talking about the possibility that octopuses respond to aversive events just with the low-key peripheral stuff. It's a surprisingly multifaceted, integrated response. There's always the possibility of asking, maybe none of this is felt. I accept that, but it shifted the contrast into that quite different place from where it was before.
Let's have a question from Claudia Passos.
Is it possible to ask a question for the previous presentation.
Ask whatever question you want.
Yes.
Yeah, so my question is for you, Jonathan. Yes, I was wondering how far you would like to go with the theory-like approach. So I was wondering if some of the theories of consciousness in the future prove that certain of those capacities doesn't correlate with consciousness. If you still think that we should not take into consideration the predictions of the theory of consciousness. So yeah, so the question is how far you think you should abandon completely the theory have approach?
Yeah, I mean, what I believe in, I suppose, is making full use of all the human evidence without prematurely committing to a specific theoretical perspective. Now, that's not intended to dismiss the importance of theories. And in fact, as we gain more and more evidence, we should be using that to build better and better theories of what the relevant distinctive kinds of processing are, and then, you know, use our improved theories to try and guide our thinking about these issues. I suppose I just think we're a very long way off the point where there'll be a sort of last theory standing. You know, we'll be here in 25 years and Dave will win his next bet. And, you know, I think we've got to be studying animal consciousness now. It's too important to say, let's leave it decades. And so I'm looking for ways to make full use of the human evidence without committing to a single theory. Yeah, so a question from the middle of the central block there. Yeah, and then might have time for one more after that. We'll see.
Yeah, following on from the discussion on opioids and the possibility that the effect was more of a reward or pleasure, that of course would be some indicative of at least a heightening of subjective experience. So I was wondering, can we devise more experiments on these little critters that would be much more pleasing and enjoyable for them to demonstrate that aspect?
Talking of little critters, I mean, again, coming back to the Cook experiment, someone says octopuses show conditioned place preference. A person might say, big deal, flatworms show conditioned place preference. And that was a be nice to the flatworm experiment. They preferred a place where, I think it was cocaine, some other drug that they were administered, and they went back to that place. So there's a tradition of being nice to flatworms, at least. And I think that reminds us, again, of the importance in the Cook experiment and work like that of the sort of, it's not just one indicator, it's the sort of multifaceted nature of the response to the event.
And I want Andrew's experiment to be a model here, hopefully, in that, I mean, what Andrew was doing in the experiment he described, there's nothing more aversive than quinine going on in that experiment. A lot of it is, you know, exploiting the bee's drive to get the sugar, you know, and they're in free-flying conditions. The more we can do of that type of research, the better, in my view.
I think it would be a better model if the results had been better. But yeah.
No, that's not methodologically. This is exemplary, and the results are the results. I think on that note, I think I'm afraid that's all we have time for. But let's thank all of our participants.
Yeah.